Good afternoon, everyone. My name is Simon Lailvaux. I'm a professor in the Department of Biological Sciences at UNO. And even though I'm going to be talking today about using machine learning, I'm not a computer scientist at all. I'm, in fact, an evolutionary physiologist. And as such, I spend a lot of time thinking about variation because genomic and phenotypic variation are the raw material that natural selection acts on to shape phenotypes in nature. And so the question that I'm really going to talk more about sort of the question and the biology around it. And what that question is sort of the holy grail of evolutionary biology, and that is connecting genomic variation to phenotypic variation. So to step back and take a very long-term historical perspective right back to basics, now there are some simple traits that do exhibit categorical variation, and you can put them into different bins, and sometimes they have a very simple genotypic basis that may be based on just one or a couple of loci, but the vast majority of traits are not like that. They show continuous variation. You can't just put them into one bin or another. They range from a spectrum. And the way that we've traditionally studied this type of variation is using quantitative genetics, which allows us to partition the phenotypic variation into additive genetic variation, other types of genetic variation, and environmental variation. And quantitative genetics is based on the infinitesimal model, which says that variation in these traits is based on an infinitely large number of genes, and each of those genes makes an infinitely small contribution to the phenotype. So in more recent years, as we've developed the ability to sequence genomes, there was this feeling that things like the GWAS, the Genome-Wide Association study, would sort of eclipse quantitative genetics. The idea was that it would bring this enormous amount of explanatory power because we'd be able for the first time to make statistical associations between specific genes and specific phenotypes. And while GWAS has been enormously valuable and has taught us a lot, it really hasn't replaced quantitative genetics. If anything, it's validated the infinitesimal model. Because even in cases where we are able to identify candidate genes or genes of large effect, we still find that most of the variation lies within those many, many, many other genes that we may know very little about. And so the problem actually comes down to the fact that the genome and the phenotype are linked through many intermediate levels of biological organization. And as you advance through those levels from the genome to the transcriptome, the proteome, et cetera, at each level, there's an increasing number or amount of complexity that you have to deal with. Even once you reach the phenotype, you still have to deal with the fact that the phenotype is acted on by the environment through the epigenome. There's symbiotic stuff like the microbiome contributing at each of these levels. And this is an enormously complex challenge, which is ideal for machine learning approaches. And so I want to talk very briefly about one model that we built to link to these levels of biological organization, just as sort of an illustration of what we think we'll be able to do. And that is a level that is very proximate to the phenotype. It is the relationship between morphology and performance. So morphology is how organisms are shaped. Performance is the kind of stuff that they do. If we're talking about animals, things like running, jumping, biting, climbing trees, flying. In some cases, we can make very direct and intuitive connections between how animals are shaped and what they can do. But if we want to do it for a whole bunch of different things, that becomes surprisingly difficult because sometimes there are clear morphological performance relationships and sometimes there aren't. What it really comes down to is we're linking the multivariate morphology to the multivariate performance phenotype. And then you have stuff like trade-offs and many-to-one mapping, and it can become surprisingly complicated. So what would be good is if we could have some kind of predictive model that could allow us to predict all of the physical things that an organism is capable of just from the shape of the animal. And this is where we started with our sort of proof of concept data set. We started with lizards because lizards are actually model organisms for performance. This particular proof of concept data set, we had more than 2,300 individuals from about 70 species of lizards, a bunch of morphological measures, a bunch of performance measures. This was just a screenshot of my laptop at the time when I was putting it together. And you can see all the red here is missing data, lots and lots of missing data. Now, computer scientists love missing data. It's like their favorite thing because whenever you have missing data, that's an opportunity to come in and build a model and do better. So the way that machine learning, I'm sure most of you are familiar with it, but very simply, you take the data that you have, you impute using some method the data that you don't have, you throw a whole bunch of algorithms and linear algebra at it, you get the answers, you compare them to data that you actually have using something like k-fold cross-validation. And if the answers that you get aren't right, you go back and you tweak the algorithm a little bit more, and you just keep doing that until the answers start looking good. So we did build our model. It works. That's always a good sign. And this is how well our model performs, right? So for these different kinds of performance abilities we wanted to predict, the model worked remarkably well. So this is how well we're doing at, with one being obviously the best at predicting these things, just with absolutely phenomenal accuracy. The reason it works so well is because we use a method called stacking, where you don't just throw one algorithm at it, you actually use a bunch of algorithms, and you feed those algorithms into each other, and you stack them on top of each other. So you get the sort of overall composite model with different algorithms producing different things. And the reason we use different algorithms for different things is that there's a theorem in computer science called no free lunch, which says that no single algorithm is good at everything. So you use a bunch of different ones, you stack them onto each other, you stack that model on top of each other, and you start being able to predict things really, really, really well. So what that means is that I can take any lizard, including any lizards you might have on you right now, and measure just a couple of simple morphological dimensions. I can tell you everything about what that animal is capable of doing. Now, that may not be very interesting to you. It's very interesting to me, but it's just an example of what we can do with these models. And so what we ultimately want to do is build a different one of these models at each level of biological organization, linking these different kinds of data so that we could ultimately have a composite model going all the way from genomic variation up until phenomic variation. So that's going to require a lot of data. And where we're aiming to get that data from is from the green anole, Anolis carolinensis. In biology, it's sometimes called the green mouse, because for about 140 years, it has been a model system in ecology and evolution. We know an enormous amount about all these different aspects of its biology, which gives us a huge amount of context for any new data we might have. It's because of this, the green anole was the first reptile to have its genome sequenced in 2011. And while that was a remarkable achievement, we now know there are a couple of issues with that genome. One of those issues is that it doesn't account for the huge amount of variation in green anoles that you see in nature. Green anoles, they range in continental North America. The history of it is they arrived from Cuba between 4 and 6 million years ago. They spend most of their time hanging out in Florida, and then about 200,000 years ago, they expand northwards to the Carolinas and to Tennessee, and then westwards along the Gulf Coast as far west as Texas. And most of the genomic data, the genome, most of the animals that it was pulled from were from this Carolinas cluster. But if you visit these populations, which I've been doing with my collaborators for the last couple of years, all these lizards are totally different in many, many different ways across the range. So there's a huge amount of genomic variation. What we aim to do is to measure all that genomic variation and as much of the phenomic variation as we can, and then build a composite model that links one of them to the other. So we've been visiting these populations over the last couple of years. We've been measuring a whole bunch, as many phenotypes as we can, and then, of course, predicting the ones with our models that we can't measure. These are a bunch of the standard sort of physiological, morphological things we have been measuring. So what is still in progress is we are measuring all of these things. It just takes a long time. It's very expensive. One of the interesting things, the new data that we've had recently is the metabolomics data. So you can take a single drop of lizard blood and identify every single biological molecule in it. That is the metabolome. This is a map of what the green anole metabolome looks like. 539 biological molecules that we identified from a drop of lizard blood. If any of you are familiar with this, please see me after. But what we're able to do with that metabolic information that is really interesting is we can recover the structure of these populations. There is signal in this metabolomic variation. We know how these populations are related genetically over time, and we can recover those relationships from the metabolomic variation, so it is meaningful. So it's still early days. We have a long way to go, but this is our ultimate goal, is to build a series of stacked models that connect different levels of biological variation and then stack those models together using all this data from the green anoles until we can eventually have a working stacked machine learning model that will allow us to go from genomic variation all the way up to phenomic variation. It's early days. We still have a long way to go, but I'm very excited about how it might work out. So thanks to all for listening. Thank you to my many collaborators. I'm happy to answer any questions if there's time.